Hello, good morning, everyone. Thank you, Brenda, for your kind introduction. So I am AGM Mostofa. I am a staff fellow in the Office of Bioequivalence. Welcome to my presentation. In this presentation, I will talk about in vitro release test, IVRT, for in situ gel or depot-forming drug products. So what do we want to learn from these presentations? So in this presentation, I will talk about some key challenges in IVRT method development for in situ gel or depot-forming drug products. I will provide some key considerations in IVRT method development and validation for this type of drug products. I will also provide an overview of the important submission contents related to IVRT study in ANDA. So let us first understand what are these in situ gel or depot-forming drug products. So these are, in simple terms, these are specialized formulations which after administration forms a gel or depot at the administration site, and that drug releases from that depot over an extended period of time. So for this drug product, the in situ form depots dictates the drug release kinetics. So far, FDA has approved 15 new drug applications and 4 abbreviated new drug applications for this type of drug product. You can see that there are relatively fewer approved generics for this type of drug products. There are multiple reasons, including challenges that are associated with bioequivalence study. This slide contains a summary of the bioequivalence study recommendation provided in the product-specific guidances of in situ gel or depot-forming drug products. As you can see that for most of these drug products, PSGs recommend in vivo BE studies. However, there are drug products for which PSGs recommend in vitro BE approach also. And in many cases, for in vitro BE approach, IVRT study is a key component. Here we have 4 examples of in situ forming drug products for which PSGs currently recommend IVRT study. Okay, now what is the purpose of IVRT study here? So IVRT study here is the component of in vitro BE approach. The other 2 components are Q1, Q2, qualitative and quantitative sameness, and Q3, which is comparable physicochemical characteristics. In case of in vitro BE approach, the bioequivalence is determined based on the totality of evidence. And here IVRT is supposed to determine or ensure that the rate and extent of drug release, which can be affected by factors such as drug substance, formulations, and manufacturing process, the comparability, make sure that the test and reference products have comparable test and rate and extent of drug release. And by ensuring that, IVRT study mitigate a potentially major bioequivalence failure mode. Now, why is it challenging to develop an adequate IVRT method for this type of drug product? First of all, there is no compendial method available for this type of drug product to follow, and the product-specific guidances do not provide any specific recommendation about the study conditions. Also, there are challenges associated with demonstration of discriminatory ability of the proposed IVRT method. However, for this drug product, the most specific challenge is optimization of the gel or depot-inducing conditions. Now, how to address these challenges? So the key to address this challenge is to understand the mechanism of depot formation and drug release for these drug products. The process of depot formation and drug release can vary from product to product. For example, there are some in situ forming drug products that are self-aggregating peptide drugs, and also there are some polymer-based formulations. In the subsequent 2 slides, I will provide illustration of the process of depot formation and drug release for these 2 types of drug products. So in case of self-aggregating peptide drugs, after administration, these peptide drugs are usually administered as solution form, and after administration, the peptide drugs undergo a self-aggregation process. And this self-aggregation process of peptides forms a dense fibrillar network, which acts as a depot. And from this depot, the peptide drug diffuses out into surrounding tissues. So for this type of drug product, the microstructure of this fibrillar network dictates the drug release kinetics. In case of polymer-based formulations, these formulations usually consist of drug substance, polymer, and organic solvent. After administration, the organic solvent diffuses out and the body fluid diffuses in. And this process causes precipitation of the polymer with entrapped drug substance. And the precipitated polymer drug acts as a depot from which the drug releases over an extended period of time by various mechanisms such as initial burst release, which is followed by diffusion facilitated release and degradation facilitated release. And for these drug products, the characteristics of the polymer dictates the release kinetics for this type of drug product. So we have discussed that for in situ gel or depot forming drug products, the in situ form depots dictates the release kinetics. And that's why, unlike the conventional IVRT method, in case of in situ gel or depot forming drug products, the IVRT method requires a gel or depot formation step. To further elaborate on this unique IVRT method parameter, which is gel or depot formation. So in case of conventional IVRT, the drug product samples are loaded as it is onto the IVRT apparatus. However, in case of in situ gel or depot forming drug products, the drug product samples to be used in the IVRT should be in a adequately formed gel or depot state. If IVRT is conducted with samples that are not in gel or depot form or not in adequately formed gel or depot, then the observed release profile will not reflect the true or actual release characteristics of the drug product. Instead, we may observe some artifacts or misleading release profiles. And this will eventually lead to incorrect assumptions on the comparability of the rate and extent of drug release between the test and reference product. So what are the key parameters for IVRT method development for this type of drug product? The parameters are sample preparation, selection of apparatus, sample loading, release medium selections, flow or stirring rate, temperature, sampling time, and specifically for this drug product, gel or depot induction. For each of these IVRT method parameter, one should take a step-by-step systematic approach for selection of the most optimal conditions. And the expectation is there should be adequate justification with supporting data for the selected conditions. And the overall objective of IVRT method development for this drug product is to achieve adequately sustained release profile and at the same time complete release within a reasonable time frame. So what are the key considerations for IVRT method development? What are the key considerations for gel or depot induction for this type of drug product? First of all, one should take an exploratory approach to investigate the factors that can affect the gel or depot formation process. Since it is not practically feasible to exactly mimic the in vivo condition, one should take an exploratory approach, explore various inducing conditions, and including the physiologically relevant conditions. And depending on the drug products, some of these factors are sample amount, the inducing media, sample to media ratio, incubation temperature, and time. And after the trial, after the induction, one should assess the consistency and reproducibility of the gel or depot that are formed using the given conditions. Next, I will talk about the key parameters for IVRT method validation. The two important parameters for IVRT method validation are robustness and discriminatory ability. In case of robustness, we want to see that slight variation in IVRT method conditions do not have significant impact on the results. And there should have pre-specified criteria for robustness. In case of discriminatory ability, we want to see that the proposed IVRT method can differentiate intentionally designed non-bioequivalent experimental test formulations. And during discriminatory ability evaluation, one should consider the critical formulation and manufacturing process attributes that can affect the depot formation and drug release. Now, what are the key IVRT study-related submission contents that we expected to see in an ANDA? The submission contents should cover these key elements: IVRT method development, IVRT method validation, analytical method validation, and pivotal IVRT study. And some typical contents are standard operating procedures or protocols that are effective at the time of a study, the detailed study reports, sufficient experimental details that will enable the assessor to reconstruct how the study was conducted, and all the data and calculations, which will enable us to assess the accuracy of the results. From agency's point of view, all these informations are critical for the assessment process. And in many instances, it is found that the deficiencies are mainly due to incomplete submission, incomplete information in the submission, which eventually delay the approval process. For awareness purposes, here we have summarized some typical IVRT study-related deficiencies that are found in an ANDA for in situ gel or depot-forming drug products. So we have found that in some cases, though gel or depot formation is needed, but the proposed IVRT method, like a depot or gel formation step, and it is not clear from the submission that the samples, the IVRT samples, are in adequately formed gel or depot. In some cases, it is found that a random condition is selected for gel or depot induction without conducting thorough exploration of different conditions, including physiologically relevant conditions. And there are some instances we found that adequate justification is supporting data were not provided for the selected conditions, or the justifications were inadequate or incomplete. We have also found examples where the gel formation was inconsistent and irreproducible, which potentially contributed to the observed high variability in the release profiles. Another typical deficiency we found is that in case of discriminatory ability evaluation of the proposed IVRT method, the critical attributes are not considered. So here are some key takeaways from this presentation. First of all, the gel or depot formation is a critical step in IVRT method development for in situ gel or depot forming drug products. It is advisable to conduct a thorough exploration of different inducing conditions, including physiologically relevant conditions. And one should aim to achieve a well-formed, consistent, and reproducible gel or depot to ensure reliable and representative in vitro release profile. For optimization of IVRT method parameters, it should be based on the understanding of the depot formation and drug release mechanism. And during discriminatory ability evaluation of the proposed IVRT method, one should consider the critical attributes which can be related to formulation and manufacturing process that can affect the gel formation and drug release. We have two challenge questions here to assess if we have achieved the learning objective of this presentation. So the first challenge question is, which of the following statements about IVRT So I'll give you some time to go through the options. And the answer is option A and C. So in case of gel or depot forming, understanding the gel or depot formation and drug release mechanism is important for IVRT method development. And in case of in situ gel or depot forming drug products, additional consideration should be given to the gel or depot forming step. Challenge question 2 is, which of the following statements about IVRT for in situ gel or depot forming drug products are false? Select all that apply. And the options are and the answer is option A and E. So in case of IVRT method, it does not necessarily need to exactly mimic the in vivo release profile. And IVRT method validation is distinct from analytical method validation, and it is expected that the submission contents should cover both aspects. So I'd like to thank Office of Bioequivalence for giving me this opportunity to talk about this topic. And my special thanks to Hisan, Utpal, and Bing from Office of Bioequivalence who helped me with this presentation and guide me with this presentation. And last but not the least, the assessment teams from Office of Bioequivalence who reviewed these complex drug products. And the basis of this presentation is those assessments. Thank you so much for your attention. Next, I would like to invite Tilok Mudaliz for his presentation.